Hello everyone, today we are going to learn about gram staining technique. It is an important staining technique clinically used as diagnostic material in the microbiology laboratory. Basically, staining simply means coloring the microorganisms or a cell structure so that it helps in learning about the microparticles. And gram stain was originally developed by Hans Christian Gram in 1884. It is a type of a differential stain. Here, differential stain means the kind of a stain that imparts different color to different organism. And in gram stain, we'll learn major about the two colors, that is purple or violet and pink. And the gram stain differentiate bacteria into two large groups, that is gram positive organism and gram negative organism. And the most accepted mechanism of gram stain is it depends upon protoplasmic pH and cell wall permeability, which will be discussed later in this video. Now we'll move on to the method of gram staining, and it requires a primary stain, which is a basic dye usually, and it may include crystal violet, gentle violet or methyl violet. Is the protoplasmic membrane of an organism is always acidic, so we are adding a primary stain as a basic dye. The second one is grams iodine, that is simply an iodine. The third one is decolorizer, that is an organic solvent, alcohol or alcohol acetone. And the fourth one is counterstain, which is again a basic dye. The usually used counterstain are safranin, carbon, neutral red, etc. So the steps involved are preparation of a smear. Here we initially prepare a smear of an organism from bacterial culture in a clean grease-free slide and is heat fixed. In step 2, we'll cover the smear with primary stain for a minute, that is crystal violet. Here both gram positive and gram negative organism uptake the dye because the dye is basic and the protoplasm of an organism is always acidic. In step 3, we'll rinse the smear with water and grams iodine for a minute. Here the iodine is a mordant which helps to intensify the primary stain. It also helps increase affinity of a stain to the biological specimen. Here the crystal violet and iodine form a complex inside the bacterial cell wall, that is CVI complex or diiodine complex, in both gram positive and gram negative organism. In fourth step, we'll rinse the smear with water and add decolorizer for 20 to 30 seconds. Usually alcohol or alcohol acetone is used as decolorizer, and this step helps to remove the CVI complex from the cell wall of an organism but not from the all. As in gram positive organism, the CVI complex is trapped inside the cell wall as it consists of thick peptidoglycan layer, but in case of gram negative organism, the cell is highly permeable as it consists of lipopolysaccharide and only thin layer of peptidoglycan. Hence the CVI complex is washed off by the decolorizer in gram negative organism. In final step, we'll wash the smear and add the counter stain for a minute. Here the counter stain usually used is safranin. Other counter stains such as carbon, neutral red can also be used. The stain that counters the color of the primary stain is usually used as the counter stain. In gram positive organism, the CVI complex is intact because the decolorizer is not able to wash off the primary stain, hence it do not uptake the safranin. But in gram negative organism, the CVI complex is washed off, hence it easily uptake the pink color of safranin. So after the step of gram staining, the final result can be visualized microscopically, and the final result shows that the gram positive organism is stained purple, that is the color of primary stain, and the gram negative organism is stained pink, that is the color of counter stain, as the primary stain is already washed off by the decolorizer. So the basic mechanism of gram staining, that is protoplasmic pH and cell membrane permeability. Now firstly, protoplasmic pH. The protoplasmic pH of gram positive organism is 2 to 3 and that of gram negative organism is 4 to 5. So gram positive organism is more acidic, hence it is able to bind more strongly with the basic primary stain than that of gram negative organism. The second mechanism, that is the permeability of the cell. Gram reaction probably depend upon the cellular permeability, as a gram positive organism is less permeable due to the intact peptidoglycan layer and hence trap the primary stain that cannot be washed off by decolorizer. In gram negative, there is high concentration of lipopolysaccharide and cell wall is more permeable and cannot resist decolorizer, finally washing off CVI complex by decolorizer, hence uptake the counter stain. So this was all about the gram staining technique, and here are the list of some gram positive cocci, gram negative cocci, gram positive rods and gram negative rods. In many cases we can get a false positive result, like the gram positive organism can appear gram negative if the smear are made from aged culture, and if there are overtime treatment of decolorizer, the cell wall of gram positive will rupture, uptaking the counter stain. Hence we need to take care of these things. So this was all about the gram staining technique, and my references are from the Microbiology and Introduction by Tota Funke and Case and the Textbook of Microbiology by Chakravarti. Thank you.